be difficult for me to give you the certificate. So be careful and be on time from tomorrow. Thank you.
Yeah, test. Yeah, it's good.
It's there, no?
Yeah, it's good. Yeah, no, I will drink.
I put few pages here. Just I. You need somebody, no?
Yeah, test. 1, 2, 3. Mic testing. Good morning. Yes, hello. All right, good morning and welcome back. Today is Thursday. We are already on the third day of the workshop. I hope you have so far gaining some knowledge, some experiences, and also a lot of interactions. And so the next 1 hour, I'm going to talk about molecular mechanisms in hypertrophic cardiomyopathy. So it's kind of how genetic variants can lead to develop the disease. Again, we talked about yesterday and day before yesterday the diagnosis, the variability, the onset, a lot of clinical aspect, but today we are going to focus on how genetic mutation in sarcomeric genes resulting in hypertrophic cardiomyopathy. Again, in a research lab, how do we study that? You know, what are the tools we are going to use? What are the models we are going to Yeah, so what are the models we are going to use in the lab and then study? So it's kind of as a scientist aspect, you know, how do we do all the experiments in the lab to test the hypothesis? Again, being a basic scientist, you know, I also do clinical study, going to the community, you know, interacting with people, and then do my research. So it's kind of, I'm going to cover a lot of aspects. So again, we went through this hypertrophic cardiomyopathy. What does it mean? You know, the phenotype, and then what are the diagnoses? The thing is that you know, when you or someone remember hypertrophic cardiomyopathy, it's a #1 cause of sudden cardiac death. That is always been reported everywhere, and people in young athletes specifically, they die if they do very severe exercise. So again, we talked about all the variabilities in the disease development, starting from the cardiac structure, the thickness, chamber size, etc., and then the penetrance. It's kind of what time in your life the disease can develop or show the symptoms, and then cardiac arrhythmias, histology, all this aspect. Then also we covered about the modifying genes, secondary risk factors, and then lifestyle choices. So these are again, we don't know how these are interacting to the disease development, and again lot of things to be learned. So the genetic of hypertrophic cardiomyopathy, again it's a pure genetic disease. Predominantly mutations are in the sarcomere gene, mainly involved in muscle formation, muscle structure, muscle function. So we are in the next 10 minutes we are going to look what are these proteins and how they work in the cardiac muscle. So this one is a real numbers, like how our cardiac system in a day-to-day activities, it's almost 72 times per minute, 100,000 times per day. That is a lot. So it's more than an engine non-stop from day one of life formation, and then before you die, this is the last organ to be failed. Again, pumping 5 to 7 liters blood in 1 minute, and then 7,600 liters per day. That's a lot. So just assume this is happening in your body, in your system every time, every minute, every second. So you can't just stop this one. So it's a very important organ. Again, you can see the coordination. Blood come into the right ventricle and then go back, and then pure get oxygenation, left ventricle, and then go out. So it's kind of constant process, but again a lot of other organs also get involved in the pumping process. If you're really going back to the history of cardiac system, we always in the ancient time, heart is always referred where we have our life. It's also been referred to character. For example, this person is a very good heart person. Somehow they related the character and the emotion to the heart. And then people believe that all our life, soul are into the heart. So that's another belief. And then it's kind of symbolic of religion. This is in 1530. It's a Lutheranism, a symbol. They introduced the heart, and then in the 17th century, it's become a love, symbol of love. So again, you can see how heart can be related to our emotion, feelings, and also character. So it's a very important organ to study anyway. This is the video kind of showing you, look at this one where we have the heart attack. So heart attack is different from sudden cardiac death. Sudden cardiac death comes through cardiac arrhythmias and hypertrophic cardiomyopathy, but this is a myocardial infarction where there is a blockage in the artery, so such a resulting lack of blood supply to the region, and so the region can die. As a result, heart can stop working. So this is called a myocardial infarction or heart attack. It's completely different than sudden cardiac arrest. Sudden cardiac arrest, people passed within a second, just stopped. Whereas myocardial infarction, they get all the pain, they feel it, they know that, but you need to go to the hospital to be treated to remove the blockage. So that's kind of the major difference. So here again, I don't know whether you can see all these letters from the heart all the way to the muscle, and then we have a single cardiomyocyte. So cardiomyocyte is the cell, single cell of the cardiac muscle. It's only in the heart, not anywhere else. Skeletal muscle, they have different cell types, but both skeletal and cardiac comes under the muscle type called striated muscles, whereas smooth muscles are different because they don't have structures like defined structures. So the cardiomyocytes, and then within the cardiomyocytes we have the myofibrils, and then you look at the myofibrils, we have the sarcomere. Sarcomere is the unit of muscle contraction. So here this is a one sarcomere from Z line to Z line, and then in the middle we have the M line. So in this sarcomere is the functional unit of the muscle. So within the sarcomere we have all the proteins like thick filament proteins, thin filament proteins, and etc., which we are going to look into it now. So if you do electron microscopic analysis of the heart, what you see is predominantly the sarcomere or the cardiomyocytes. Within cardiomyocytes you have the sarcomere, which is surrounded by mitochondria. So again, mitochondria gives the energy for the muscle function, whereas the sarcomere generate the forces. So again you can see this is the Z line to Z line, it's called one sarcomere. In the middle we have the M line. And the dark line, they are the thick filaments. In between two thick filaments we have the thin filament proteins. Again within the heart, 75% of the cell types are cardiomyocytes. But we also have endothelial cell, fibroblast, all other cells within the myocardium. So again this is the schematic diagram of the sarcomere. You have thick filament, titin, and then myosin heavy chain, and then the myosin binding protein C, and then thin filament proteins, the troponin complex, actin, alpha tropomyosin. So these are the major sarcomeric proteins, and then the myosin heavy chain has two light chains, regulatory light chain and essential light chain, and those are attached to myosin heavy chain. Again myosin is the motor protein, we talked about that yesterday. So again if you look at how this contractile property occurs, a lot of signaling events can alter the force, the speed, all the property of contractility can be modified through so many factors which are listed here. For example, beta adrenergic stimulation, such as activation of catecholamine. You can see all these pathways can get activated, the kinases can be activated, resulting that they can directly come to the sarcomere and then activate or inactivate the function of the muscle. So today companies are focusing on instead of going through all the pathways, can we directly target sarcomeric proteins and then treat the disease hypertrophic cardiomyopathy. So again sarcomere is the contractile unit. We have thick and thin filament proteins, so you can see how they are. So thin filament proteins primarily actin, and then we have the troponin complex, and then alpha tropomyosin. So within troponin complex we have three major protein: troponin I, troponin T, and troponin C. Troponin C is the calcium binding protein, the C indicates that, and when calcium binds it releases the blockage, so that myosin can directly interact with actin and force can be generated. And then the thick filament proteins, primarily myosin, and also we have titin, and then myosin binding protein C. Titin is the largest molecule. It runs through from Z line to Z line. It really kind of anchoring, holding the entire sarcomere. Major protein. If mutations in titin mostly related to dilated cardiomyopathy. So this is the standard. If you look at any biochemistry book, how muscle works, this is a kind of cross-bridge cycling. So in this one is very every beat, every contractility, you can see ATP comes in, binds to myosin, and then same time calcium binds to the troponin C. So then once that binds, the myosin is now exposed to interact with the actin. As a result, the power stroke can be formed, and then the ATP at this time binds, and then the hydrolysis, and then it releases the ADP. So it's kind of circle every beat we do in the heart. So again, if there is any change in this cycle in the amount of ATP, the amount of calcium, that can alter cardiac function. So I put this one just for the students level, how this work. It's like our car engine. The sarcomeres, the muscles are very much builder in a way. Everything should be in a proper position, location, and the distance between two sarcomere in the myosin heavy chain, where they arrange myosin binding protein C. For example, every 11 myosin heavy chain has one myosin binding protein C. The distance between two myosin binding protein C is 43 nanometer. So everything is very much defined in the muscle. So you cannot just mess up that one. So again, you can see when calcium comes in, then it binds to actin, and then force can be generated. So this is a very defined structure. You cannot mess up the system. Once you started messing up, and then the disease or any changes in the function can be occurred. So here is the structure of these two major protein, myosin heavy chain and myosin binding protein C. This is the motor protein. We have myosin is always like a heterodimer. They always have in two different protein. So the head region, and then we have the proximal S2 region, and then the light meromyosin region. Whereas myosin binding protein C, change it to laser. Myosin binding protein C, you can see here, it's also very large protein, 150 kDa protein. At the N terminus, you have C0 domain, and then all the way to C10 domain. And again, there are various functionality. For example, in this slide, you can see the C terminus of myosin binding protein C interacts with the myosin heavy chain, whereas the N terminus reaches to actin as well as myosin heavy chain. So this interaction is so critical for regulating the amount of force myosin heavy chain can generate. We call it myosin binding protein C as a kind of switch based on how much inhibition or activation occurs, the force can be generated. Right now the light switch, I don't know whether you have here, you can increase or decrease the amount of power, the electric power. Based on that, you can have more brightness. Similar way, myosin binding protein C works against myosin heavy chain. The more attachment, the more binding to myosin, less force, the more free away from myosin heavy chain, we develop more force. So going back, so this is the definition of hypertrophic cardiomyopathy. So it is called disease of the sarcomere. Why? Predominantly mutations in the sarcomere genes are linked to develop the disease. That's why this has been named as the disease of the sarcomere. Again, these are the major sarcomeric proteins we have. and top two are beta myosin heavy chain and myosin binding protein C. And again, if we have the mutation in these two genes, it's almost a guarantee to develop the disease. So how the sarcomere dysfunction occur? So here we have the normal sarcomere. Again, you can see the myosin heavy chain attachment to actin. Normal contractility, effective relaxation, they are very ordered in the sarcomere structure. What happened when we have dysfunction because of hypertrophic cardiomyopathy? You can see there are more myosin heavy chain attached to actin. That is called hypercontraction. So the more attachment, we generate more force. So again, hypercontraction because more actomyosin interactions, and then impaired relaxation because the cardiac tissue has very strong stiffness, and then we develop the fibrosis. So this is all part of the the hypertrophic cardiomyopathy disease process. As of today, again we have over 1400 different defined mutations that are directly associated with development of the disease hypertrophic cardiomyopathy, and 11 or more genes are now known. Again, mostly are in the sarcomere, and two most common genes are the myosin heavy chain and myosin binding protein C. This is one of the paper we reported that we went to a local hospital, and they again in the States we have genetic screening is routine. When they identify a hypertrophic cardiomyopathy patient, they immediately do genetic screening. So we have a panel of Illumina DNA sequencing system. I think it's around $2,000. The cardiology refers them and they do the sequencing, and so we got all the data set of the families. I think we had 126 families, something like that in the study. Of that, we identified 18 genes and then 125 mutations in 14 genes. Of that, 50% from myosin binding protein C and 24% from myosin heavy chain. Again, myosin binding protein C has more number of mutations. So myosin binding protein C, again interestingly 85% of the mutations in myosin binding protein C are truncations. What does that mean? You have a stop codon in the middle of the gene, resulting that you are not producing the whole entire protein. So only half of the protein you are producing, and that could cause the hypertrophic cardiomyopathy disease. And then 70% of the mutations are linked with non-obstructive hypertrophic cardiomyopathy phenotype. Again, there are two major potential mechanisms. One is haploinsufficiency, and the second one is Poisson polypeptide. So haploinsufficiency is nothing but you have two alleles. One is a mutant allele where you have a truncated myosin binding protein C, and the truncated myosin binding protein C never been exist in the sarcomere because it cannot bind in the sarcomere, so it's been removed by the ubiquitination proteasomal system. So resulting you are living with only one allele and you have normal protein. But is it enough? The protein you generate from the wild type allele, is it enough to maintain normal sarcomere function? It is not. That is called haploinsufficiency. So again, when you undergo any workload or developing aging process, the one allele is not enough to give you 100% of the protein. So again, that's kind of called haploinsufficiency. The second one is the Poisson polypeptide. I will talk about some of our experiments on that one. And again, you can express the mutant protein and the mutant protein can go randomly binds in the sarcomere and then cause contractile dysfunction. So, now I am going into some background about what is myosin binding protein C and what are the phenotype characteristic. So, myosin binding protein C, we have 3 different paralogs. Paralogs are not isoforms. Paralogs are unique genes. The protein can come from their own gene. Isoforms are from the same gene, but splicing, alter splicing can cause the isoforms. So, here we have 3 different myosin binding protein C. Slow skeletal myosin binding protein C encoded in chromosome 12, fast skeletal myosin binding protein C encoded in chromosome 19, and then cardiac in chromosome 11. As you can see, they are like very close, similar domain structures. The round shape or the immunoglobulin domain, and this one is the fibronectin domain. So, cardiac has extra 100 amino acid C0 domain, and then a couple of phosphorylation sites, and then there is a unique insert within C5 domain. But mutations in skeletal myosin binding protein C can cause skeletal disease, not cardiac disease, because they do not express in the cardiomyocytes. So, why we have to study myosin binding protein C? Again, it is an important protein, it is a transfilament protein, it connects thin and thick filaments. Without the myosin binding protein C, sarcomere structures are altered and they do not be normal in shape. And you can see it is also heart break, it regulates the actomyosin interaction and the rate of force development. Again, if you look at the electron microscopic analysis, myosin binding protein C is very specifically localized within the Z zone of the sarcomere. So, again there are 7 to 11 stripes. Every 11 myosin heavy chain has 1 myosin binding protein C. As I said, the distance between 2 myosin binding protein C is 43 nanometer. So, it is very defined structures in the sarcomere. What happens if you remove myosin binding protein C by knockout? So, this is a mouse heart and wild type 3 months normal, whereas this is a heterozygous. One allele is completely removed, another allele we have, and here we have both alleles are gone. You can see there is a hypertrophic phenotype within 3 months if you do not have myosin binding protein C. So, what happened when you knock out myosin heavy chain? It is embryonically lethal. What happened if you knock out troponin I? Embryonically lethal. But when you remove myosin binding protein C, you still form the heart function, but they develop severe phenotype, failing heart down the road. And also myosin binding protein C has more number of genetic mutations than any other genes right now. Okay, so again we talked about 2 different mechanisms or mutations. One is gain of function, another one is loss of function. So, gain of function means more than normal function, loss of function is less than normal function. So, pretty much these are the 2 major examples. In myosin heavy chain, predominantly most of the mutations resulting in gain of function. What does that mean? Myosin is now being hyperactive, more active in utilizing ATP and hydrolyzing ATP and resulting generating more force. In myosin binding protein C, when you have the mutation, it causes decreased force and then decreased ATPase activity and decreased interaction with other proteins. So, this can be a simple example how to define these 2 different gain of or loss of function mutations. So, looking into myosin binding protein C, again I said 70% of the mutations are truncations, they have in-frame insertion or deletion or splice site donor or acceptor site mutation, or premature termination, or insertion or deletion in random intronic region or exonic regions. And then only 30% are the missense mutation, where you have the full length myosin binding protein C. So again, hypothesis both in mouse models and also in vitro studies, this can be various mechanisms can be found on the literature. For example, some mutations either in troponin or troponin T, for example, can cause high calcium sensitivity. Some mutations can cause poison polypeptide. Some genetic variants can activate more inflammation. Some mutation can cause direct energetic deficit. And then we talked about loss of function, gain of function, haploinsufficiency, low calcium sensitive. So this all can be potential mechanisms for developing hypertrophic cardiomyopathy. So one of our graduate student, David Barfield, he studied haploinsufficiency using a mouse model, where we have one allele is a wild type normal, another allele is completely mutant allele, and resulting there is no protein. So the model we have is just one allele, but showing a very severe phenotype. The poison polypeptide is another one which I am going to present right now. It is kind of my PhD work, but we still continue the project. You can see when I started the project and where we stand on right now. So before I go into our research project, I wanted to make sure you have any questions so I can answer them, and before I jump into my research project. So far, do you have any questions for me? I don't know exactly the number. It definitely vary between animals, humans, so it's also the aging process. Mostly we look for compared to the control versus mutant, but again I don't know the number top of my head. Princess, do you know the number? I don't know how much micrometer. 2.5 micrometer Z line to Z line.
I have one more question. Sir, you mentioned in your presentation that myosin C3 and mentioned that 80% of the mutations are truncation. Is this the only protein that where this 85% the mutations are truncation? Are there any proteins reported in the literature where percentage mutation of truncation is that high?
Yeah, for example, titin, right? Titin, the giant protein, that has also lot of truncation, but the disease is dilated cardiomyopathy, not hypertrophic cardiomyopathy.
And a following question of the same, when you say that condition is the stop codon has been inserted, something like that. So in-frame deletion, addition is not likely to lead to the generation of stop codon. Point mutation is likely the reason to introduce this stop codon.
So in frame, so if you, for example, if you introduce one nucleotide, that changes the codon, right? You just move the codon, right? And then you can introduce stop codon. Or if you delete one nucleotide, that changes the in frame every 3 nucleotide, right? That can also introduce stop codon.
There was an image shown by you about the micrographs of the sarcomere structures. They were generated using EM.
The one picture, the
Sarcomere.
yeah, that's EM, electron microscope, yeah, TEM, yeah. Yeah, please.
Cryo.
No, standard electron microscopic picture. Yeah, no. Yeah, I mean, if you look all the sarcomere literature, this is the one immunogold electron, sorry. This is the one immunogold electron microscopic analysis where you stain the sections with antibodies and then you have a gold, I think immunogold staining. Then you can identify the striations. The other one this one is a very standard electron microscopic cross sections, and you take an image and then you will see it. You don't need to have any specialized technique for this one. Yeah, this is you can, it's in the standard textbook, you can find this one.
The role of calcium in this case, generally calcium functions as a second messenger.
Yeah.
So is this the way that calcium binding is playing the role with the myosin binding protein?
Yeah, so when you alter contractile function, the uptake and then release, or we call it the on rate or off rate of calcium binding, right? That can change. Resulting, you can have more calcium in the sarcomere that not been utilized or they've been more utilized. So that could cause cardiac arrhythmia, you know. So we call it myofilament calcium sensitivity. We study how much calcium the myofilament required to generate off maximal force. You know, 50% of the force, how much calcium required. Comparing to the control, you can see. So hypertrophic cardiomyopathy mostly hypercalcium sensitivity. We can also use ion optic system where you can freshly isolate cardiomyocytes, and then you can also study the calcium transients by using a very specific calcium labelings. You can see how fast calcium comes and then goes from the sarca, where calciums are stored within the sarcomere. Again, whatever you alter, you know, based on you know, the condition, that can change amount of calcium in the sarcomere.
When you say haploinsufficiency, so does it mean that one of the SNP which is responsible for the disease manifestation is either missing or is less in frequency? What does that actually means?
Yeah, so I will go through the next set of real experiment in our lab. So you have two alleles, right? Haploinsufficiency comes when you have a truncation, you don't have the full length protein, you have half of the protein, and this half of the protein is not properly localized in the sarcomere. So they are always floating around. That can be removed by the UPS system. So you don't have this protein at all, right? So now you have only one allele, the wild type, and you generate whatever that allele gives you, mRNA and protein, right? So you don't have two alleles now, you have only one allele. During the aging process, the amount of RNA and protein from one allele is not enough to maintain normal structure. Yeah.
So this is in particular about one particular gene or about any gene?
Any gene, if the protein is not present or not incorporated normally, and so you just have only, so for example, heterozygous knockout mice, any gene you knock out and you have the heterozygous, that could be a haploinsufficiency.
So just a basic question. So You mentioned a table where you talked about the two proteins, myosin and the C protein, that in one protein if there is a mutation, those are mostly gain in function. Other one it is loss in function, right? So if a C protein is named like a brake for like muscle contraction, so if there is a mutation in the brake, it could be a gain in function or.
So it depends, right? So if you have a haploinsufficiency, now your myosin heavy chain has no brake, right? No control by C protein. It's kind of phosphorylation of C protein. So if you don't have C protein to control actomyosin interaction, so you have more hypercontraction. So that's a gain of function of the myosin heavy chain, but not myosin binding protein C, right? You don't have myosin binding protein C. Yeah. Okay, so the next set of talk, I'm going to give an example of a project that we do last 20 years. So that's going to be very interesting. So in myosin binding protein C, there are several founder mutations being reported in the literature. Again, I listed a number of different mutations. What is founder mutation? It is a mutation that is very specific to very specific ethnic group or a population or from a region. You know, that can be a founder mutation. For example, the one from Germany, this 702, this one is exists over 50 generation in a region. So that's been a founder mutation. So we are focusing on now a 25 base pair deletion in intron 32 of myosin binding protein C, and that was my PhD discovery, and that's what I'm going to talk about right now. So the variant is 25 base pair deletion at the branch point exactly within the intron 32. That could result in skipping exon 33 or altered splicing of exon 33. So it's not like giving you a normal splicing. So resulting that you are modifying the C10 domain of the myosin binding protein C. So this region is so important for the protein to be properly localized within the sarcomere. If you modify this region, the protein cannot be present in the sarcomere. It will move away, it will be removed by the ubiquitination system. So followed by my work, PhD work, this is Dandapani, he is at now IISc Bangalore. He extended the study and then he collected samples from the rest of the world looking for this 25 base pair deletion. So from my study, we only focused on South India from Tamil Nadu and Kerala, but then this study extended and we got a paper in Nature Genetic in 2009. And you can see the yellow spot, that's the region the 25 base pair deletion is located or present. It's almost 4% of the population carry this variant. Again, if you based on today's population, we expect over 100 million people can live with this variant. And then the problem is not a problem, 95% of the people with this variant are normal or incomplete penetrance. And if you have it and if you see the disease, predominantly it's a late onset. After 50 years, 60 years, you see the disease difference. So we kind of concluded in my study and his study that this can be a secondary risk factor. Again, we talked about hypertension, diabetic, etc. So if you have this variant plus all those secondary risk factors, you can develop the disease early on. But the question was like, how exactly the 25 base pair deletion caused the disease, and is it really caused the disease or not? We don't know. So that's what, when I started my own independent lab, we thought like, okay, we have to revisit this study. So in the US, South Asians, people from India, Pakistan, Bangladesh, Sri Lanka, so almost like 5.4 million people. I was at Loyola University of Chicago, and I looked at the number in Chicago. It's almost 400,000 people living. So in my neighborhood, so if I say 4% of the 400,000 people, I have 17,000 people just in Chicago walking with this variant. So I don't need to come all the way here to do any genetic study. So I thought, okay. Then I had a summer student. This guy is Aravind Jagadishan. He was graduated from undergraduate and then joined my lab, and he wanted to do a research project. So I told him like, we need to go to our community, collect samples to screen for the variant. So he named the study called Diastolic Dysfunction of South Asians, or DOSA study. As you know, DOSA is so popular. So when you put this kind of title, people get attracted, they come, they're curious, what is this? And so we can easily convince them to collect saliva samples or blood samples. So he went around, and so here are the region or cities. Wherever there is a Diwali event, Pongal event, any festival, we go there. We put the booth with the consent form, everything, and so we started collecting samples. So in the study we reported in 2018, we collected sample 2,401 samples we collected, we screened. Of that, 139 heterozygous, and then homozygous 5. So it was very clear that it follows the polymorphic variant definition. So again, you can see we have enough subject carrying the variant, and also we have a homozygous subject. So this is even more than what we reported from here. 6% of the population, this one carry the variant. So the question is, again in this study, we invited these genetic carriers to the hospital. We performed echocardiogram. At baseline echo, again 95% of the subject carrying the mutation were completely normal, which was very good news for them. And again, you are wondering why then we have this variant, what is happening? But the 5% of the subject carrying the variant show very severe phenotype, and they had hypertension, they had diabetic, and they were aged people. So we thought like maybe if you are 40 years, 45 years, you have the phenotype, but it's not enough to show by normal echo. So we then brought them back again to come to the hospital, where we performed, this is called bicycle stress echocardiography. You can see we had to have a cardiologist in the room, we had to have a sonographer, 2 assistants, and then my postdoc. So just to do one study, we had to coordinate so many things. And so this was started 2019, just 1 year before COVID. So again, we invited the 25 base pair deletion carriers, 10 subject with the genetic variant and 10 subject without kind of normal. We did whole genome sequence. We excluded any other genetic variants in this subject, and then we had only 25 base pair deletion. So you can see very nicely at the baseline there is no difference between the mutant and the wild type or non-carriers in cardiac function. But when you increase the stress, giving more stress stage, you can see there is a clear difference in their cardiac function. That tells that at subcellular level, you have the phenotype, but it's not enough to show at the organ level, bigger organ level in a normal baseline condition. You need extra stress to show that, you know. So, this was a striking study. So, then we have made the mouse model just to overexpress the mutant protein, you know. So, what happened if you express the mutant protein? The answer was, again I showed this slide yesterday, the mouse with the mutant protein, they develop clear hypertrophic cardiomyopathy phenotype in 3 to 6 months of age. You can see they have the hypercontraction, the ejection fraction is increased, fractional shortening is increased, they have more thicker ventricular wall and small chamber size, etc. We had 2 papers, one is in Journal of Biological Chemistry, another one is Cardiovascular Research, talked about the pathogenicity of this variant. For example, here, so when we express the wild type protein and the mutant protein, so here we have a control, you know, this is a cell culture experiment, uninfected and then the empty vector. So, we express a normal level of these 2 proteins, right. And then when you solubilize the total protein from the cardiomyocytes, you can see the soluble fractions, we have more mutant protein, the wild type is very, very less. Again, MIG-TAG is a kind of extra 10 amino acid we introduced at the N terminus of both wild type and mutant, just to show the difference from endogenous versus exogenous. Lastly, in the myofilament fraction, so myofilament fraction is the non-soluble fraction, you know, where all the proteins are intact, where you can see clearly that the wild type is only present, whereas the mutant protein is almost very minimum, meaning that the mutant protein is not incorporated, it is not binding in the sarcomere, it is expressed, it is there, but not in a proper position or location. So, again this could be a Poisson polypeptide effect. So, when you do confocal image with this specific antibodies to see where this mutant protein localized in the sarcomere, you can see compared to uninfected control, the wild type is very normally, you can see the doublets, very normal incorporation, whereas the mutant protein is distributed, diffused within the sarcomere. So, again this could cause severe phenotype development because the muscle cannot properly function. Now we have this organoid, we have now iPSC derived cardiomyocytes from patients or from subject carrying the variant, and we can convert them into small spherical organ like structure. In one Petri dish we can have 36 organoids, so you can really multi-scale and you can do various studies like a drug screening or compound testing or phosphorylation testing, anything you want to screen you can use this model, you can measure the function, everything. Again we have a paper this last year published just to establish the technique, and we can use this model to study hypertrophic cardiomyopathy. Any questions so far before I go into what we are doing right now?
Sir, I have one question. Are you saying that this 25 base pair deletion is a founder mutation in the population of South Asia?
Correct. Correct, it is.
So the question is like, can we check that if this 25 base pair deletion is evolving with the evolution of human being? Like, can we talk about the selection pressure on this particular mutation in the population? Like, if the selection pressure is, you can say, if it is helping or if it is beneficial.
Yeah.
I know it is causing the phenotype, disease phenotype, but if it is being selected, then there is always a chance that in future we will have more burden of the disease. So can we check on those lines?
So we really look back at what time in the evolution this 25 base pair. because
So, sir, sorry. So that can be checked by the allele frequency which is there in the population, whether it is already established itself in the population
yeah,
or.
we have established, so again I want to go back, we determined based on the SNPs and the age, like at what
The age of the-
you know, in the evolution, around 25,000 to 35,000 years ago. I think after we migrated from Africa, someone got it and then distributed. The reason why I'm saying is, from Pakistan to all the way to Kanyakumari, the distribution is the same across all the religion, all the community, all the caste system. There is no difference. It is there 4%.
Yeah.
It's not been very selective in one population or one community or one caste system.
No, but all the Pakistan and everyone, we call us as a Asian population.
Yeah.
So if the selection is happening for that, then everyone, whosoever comes in this region of the world, they are under that pressure.
Correct.
So.
Yeah. So the selection process, I don't know whether there is any beneficial, why we had to carry this variant this many years, if it is not beneficial, right? We don't know the answer. Again, it's kind of, you can imagine, we have to do a very systematic study to answer that.
Now, why I'm asking, now we have the 1,000 genome sequencing database.
Yes.
So if it can be exploited to answer that question.
Probably, yeah. Maybe that's a great project for a student here. What do you think?
I don't know. I just shared my view. Like,
Really?
if
Yeah,
it can
no,
be.
I mean, it's a computer, I mean, they can also look all other, the UK Biobank
Yeah,
database,
exactly.
all other database, put together a nice study, yeah. Yeah, great suggestion, yeah.
Thank you.
Yeah. I will finish it in 15 minutes. Any other questions? So again, you can see I started this project 1996. So we are still going on without knowing exactly what this mutation is doing in our population. So the animal model I showed was a transgenic model. We used alpha myosin heavy chain promoter to overexpress the mutant protein, and so again we wanted to understand exactly the mechanism and how this 25 base pair deletion in intronic region, not within exon, intronic region caused the disease. So I moved a year ago to Arizona. So again you can see a lot of cactus. It's a very nice place to visit. I recommend strongly to come and visit me. So we have now two different models. So we created a humanized mouse model in which we removed the intron of mouse and we put the human intron with or without the 25 base pair deletion. So then we can see really the splicing, you know, the RNA expression, then also the phenotype presence. So you can see in the DNA sequence here, this is a splice branch point. The deletion is here in the polypyrimidine track is almost completely knocked down. So which is a critical region for splicing. So what we found, so again within, so here we have the cardiac function, and there is a clear phenotype in these mice. They develop heart failure, they show hypertrophic phenotype, and so the mutation either you overexpress or have introduced as knock-in, you can see a phenotype. So meaning that this is the genetic variant with pathogenesis. We also looked into challenging these mice to the catecholamines. stress and see whether they show more cardiac arrhythmias compared to wild type control, and the answer is yes, we do see a lot of the PVCs and also the NSVT, non-sustained ventricular tachycardia. So again, these mice are now been characterized by my another postdoc. We look for what exactly at the splice donor and acceptor site, how the deletion is causing the mutant protein. So in normal splicing, we have the splice donor and then splice acceptor site, and this is the region we have the mutation. And in that region, you can see there are so many transcriptional factors that are involved in like binding and then transcribing normal mRNA. The RNA binding motif proteins are key proteins. You have to have normal binding and then post-translation modifications. So again, in the literature, you can see various alter splicing in heart failure, in any kind of disease process. Pre-mRNA, you can have a normal splicing, and then you can see complete exon skipping, alter splicing, alter 3 prime, this is alter 5 prime, intron retention, and then mutually exclusive exons. So again, these are the various types of alter splicing. And again, we know that from the literature, if anything changes in these, and then also mouse models with alter the RNA binding proteins can also lead to develop heart failure. So we then look into the myosin binding protein C, again using the database, how many genetic variants been reported in the literature on the splice acceptor and splice donor side. Clearly there are plenty of mutations. So again, it's an important regions to study. Here coming back to the 25 base pair deletion, this is an in vitro experiment where we used exon trapping experiment. So we cloned the region with the intron and then exon 32, then intron 32 with or without the variant. It's a in vitro experiment using exon trapping plasmid construct system. H9C2 cells was used, we transfect, and then after 48 hours we collected mRNA and then look for the alter splicing. So clearly the 25 base pair deletion causes alter splicing, it's guaranteed, it's there. Also in the mouse, so we go back to the knock-in mice, we also see there is a clear splicing, alter splicing present also in the mouse model carrying the 25 base pair deletion. So this is an RNA-seq. Again, you can see the splicing, all the potential splicing occurs with the 25 base pair deletion. Here we have the normal splicing. So definitely the 25 base pair deletion causes the alter splicing. And then we also looked on the level of expression of RNA binding motif proteins. There are 3 major different RNA binding RBM proteins, RBM 20, 24, 38, and they are upregulated. So the more RNA binding protein present, the more alter mRNA can be present. And that's what we do see in our long RNA-seq. So again, we confirm that by doing the long RNA-seq, there are increased alter splicing in these mouse models. Currently what we are doing is now is that we have iPSCs from human subject. I showed you the organoids, we have the mouse model, and the idea is right now we are developing antisense oligos against the mutant or alter mRNA, and that can be introduced into either the cardiomyocytes or animals, and that can be used to remove the mutant mRNA as a beneficial effect, and that's kind of we are now moving forward. We have a pattern, a provisional pattern, and the full pattern is pending right now. and we have been working on this system for now. With that, I want to stop right now and then take you any questions. The last part. So again, we have the 25 base pair deletion, intronic mutation, cause alter splicing, but again, lot of question we are still asking whether how these secondary risk factors like hypertension or diabetic, will they increase the RNA binding protein motif expression that could cause more alter splicing, or aging process, for example, is it aging can also increases this RNA binding motif proteins expression level that resulting in more altered mRNA of the myosin binding protein C. So, we are looking so many secondary risk factor associations. Again, we try to answer so many questions still. Again, this is Arizona. We do a lot of hiking, mountains, so much things we can do other than just work in the lab. Okay, any questions before I conclude? And then we have our next speaker is here. Okay, we can talk during the coffee or break or bio break time. Thank you.
Thank you so much, sir, for such informative talk with so much of illustrations, and we definitely something we understood about sarcomeric proteins. And before we start with our next lecture, I welcome on the stage our head of the Department of Zoology, Professor Narahari Gramapurohit, sir, our course coordinator, Professor Varsha Wankhede, and our distinguished cardiologist, Dr. Abhinav Mohabey on the stage, please. Before we start our session, it is my honor to introduce our distinguished guest and cardiologist, Dr. Abhinav Mohabey. Dr. Abhinav Mohabey is a highly accomplished cardiologist with a strong academic and clinical background. He completed his MBBS from Dr. Punjab Rao alias Bhausaheb Memorial Medical College, Amravati, followed by a DNB in internal medicine at Max Super Specialty Hospital, Delhi. He went on to pursue his DM in cardiology from Bharati Vidyapeeth Deemed University, Pune, and is currently undergoing a fellowship in cardiac electrophysiology at Ruby Hall Clinic, Pune. Professionally, Dr. Mohabey has served as a senior resident in medicine at Babu Jagjeevan Hospital, Delhi, and Chandulal Chandrakar Memorial Medical College, Bhilai. His clinical expertise includes performing and assisting in over 1,500 diagnostic catheterizations, more than 100 coronary angioplasties, and several device implantations, including pacemakers, AICDs, and CRTs. In addition to his clinical work, he has presented in multiple papers at prestigious national conferences covering rare and challenging cases in medicine and cardiology. With this, I request our head of the department, Professor Narahari Gramapurohit, sir, Professor Sakthivel Sadayappan, sir, and course coordinator, Professor Varsha Wankhede, to welcome and felicitate Dr. Abhinav Mohabey. Thank you. So now I request Dr. Abhinav Mohabey to come and grace us with his inspiring talk and speech.
Good morning, everyone. Thank you very much for kind It's not coming here. Am I audible? Or should I directly speak?
No,
Project होएगा ना? Should I duplicate it? Okay.
काढून परत लावा डीएम आहे ना हो डीएम आहे हो डुप्लिकेट करायला लागेल ना डिस्प्ले डुप्लिकेट करून डुप्लिकेट करून सर वो लैपटॉप लगाना वो लगाना है
You have this one? Yeah. Yes. Meanwhile, yeah. Good morning, everyone. Thank you very much for kind introduction and kind words. So, I am very grateful and I am very thankful for the Department of Zoology in this university, and I am very grateful to HOD sir as well as Varsha ma'am for giving me this opportunity to speak in this platform. So, I am here to discuss management of hypertrophic cardiomyopathy. So, I hope till now, right now I will be discussing the phenotype. So, the previous lecture was more on genetical aspect. Okay. So, I will discuss little bit of hemodynamics as well as basic of physiology as well as pathophysiology of hypertrophic cardiomyopathy. I hope it has been covered in previous lectures, but for understanding the management and for understanding the drugs as well as how they work, how the mechanisms of those drugs help in inhibiting or controlling the symptoms as well as giving us mortality benefit. So, for understanding all those things, we need to understand the mechanism, what is the normal physiology and what is the abnormal physiology and pathology which get developed in this diseased state. So, basically in this slide I would like to cover basic physiology as well as pathology. So, I will start with normal physiology. So, this part, this is normal LV, that is left ventricle, this is left atrium. Okay. This is left atrium, left ventricle, these are mitral valves, these are anterior mitral leaflet, posterior mitral leaflet. Then we can see this is LV cavity, then this is LV outflow tract, this is aortic root, aortic cusps, then this is sinus of Valsalva, and this is arch of aorta. Okay. So, why I am giving so much of stress in this normal and basic structure, which is very normal in our day-to-day activities, because for understanding pathology we should understand physiology. That's how we'll be able to understand the symptoms with which patient present, and that's how we can able to jot down the treatment as well as what are the mortality benefit patient is giving from their treatment, and why they are getting that mortality benefit, and what will be the ultimate benefit of all these things, like mortality benefit, recurrent hospitalization because of failure. Okay. So, again I will come to the basic physiology. Okay. So, as we know, cardiac cycle is composed of diastole as well as systole. Diastole means during diastole LV will relax. Okay. LV means left ventricle will relax, and blood will come from left atrium to left ventricle, and mitral valve will get open, just like this. So, initially at the end, at the start of diastole, blood will be in left atrium, then mitral valve will open because of pressure difference, and during diastolic filling phase, blood will come from LA to LV. Okay. So, it get filled here. So, this is end of diastole. I am not going into too much of detail, because otherwise it will be like the whole day will be less to discuss everything, but just the gist to understand the concepts. So, during diastole LV will relax. So, because LV will relax, so there will be negative pressure here and positive pressure will be here because blood is there. So, because of this pressure difference, mitral valve will open and then blood will enter from LA to LV. So, at the end of when blood will enter to LV, it will be the end of diastole. Now, systole will start. Systole means there will be contraction. LV will contract and blood will pump into the subsequent arteries, from RV into the pulmonary artery, from LV into the aorta. So, here I am just focusing upon left part, LA, LV. So, at the start of systole, what happens? There will be isovolumetric contraction phase, because aortic pressures are high and LV pressure to start with are low, 30 or 20. So, during isovolumetric contraction phase, LV will contract. Okay. Why it will contract? Because these are LV, these are wall of LV, this is LV cavity, and wall is made up of myocytes and muscles, and inside muscles there are all those filament and proteins are there, about whose mutation we were discussing in previous lecture: actin, myosin, troponin, titin, tropomyosin, everything. So, this is how like I want to like correlate everything. So, if you will do the microscopic cut section and see, so everything is here. So, multiple fibers of myocytes accumulate to form whole wall. Okay. So, when it will contract because of actin myosin contraction, when ultimately group of myofibril will contract, so group of muscle will contract, it will create a force, and because of that pressure will increase, and once pressure will increase, this anterior mitral leaflet and this posterior mitral leaflet, they both will come together, it will close, and after that what will happen? This aortic valve will open once the pressure in LV cavity is more than aortic pressure, approximately 120. We know our blood pressure is 120, approximately 120 by 80. So, this aortic pressure will be 120. So, once by contraction of these muscles, actin myosin filament, when LV pressure will be more than 120, so the aortic valve will open and blood will enter into aorta. So, this is normal physiology. So, we can see this normal wall and normal cavity. Now, coming to hypertrophic cardiomyopathy, as we know, it's a pathology. Okay, it's a pathological condition. Predominantly, it is an autosomal dominant. Okay. So, most of the means there can be other comorbidities and contributing factor, but predominantly it is because of genetic mutation and different gene, okay, leading to deformation of protein, leading to deformation of that particular myocytes. That's why what will happen, because of this genetic mutation, the arrangement of protein will get changed, and if you will see in microscope, there will be myocytic disarray. The matrix deposition will be disturbed, it will be distorted. The protein metabolism and mechanism and energetics will get changed. Because of that, what will happen? There will be hypertrophy. As the name itself suggests, hypertrophic means large, cardiomyopathy, cardio means muscle, cardiac muscles, and myopathy means disease of the muscle. So, this is a disease of cardiac muscle in which it will get enlarged and dysfunctional. So, that is why so depending upon which group of muscles are involved, we can have different types of cardiomyopathy. I am not going to detail because I suppose it has been covered in previous lectures, but just for understanding basic I am just revising it. Okay. So, this is the classical form. So, this is septum part, this is posterior wall, this is septum, this is posterior wall, this is normal heart, this is hypertrophic. Why? Because here we can see there is an elevation here. So, this is because of mutation, because of disarray, because of matrix deposition. Okay. So, there is a hypertrophy, we call this hypertrophy. So, this is asymmetrical, this wall is normal just like normal, but here it is elevated, hypertrophied. So, this is asymmetrical septal hypertrophy, okay, which is the most common presentation. Depending upon which group of muscles are involved, it can be, see this LV can be divided into base, this upper 1/3 is LV base, lower middle 1/3 is mid part, and lowermost 1/3 is apex. So, this part is apex, this part is mid LV cavity, this is basal. So, depending upon which sets of groups of muscles are involved, it can be apical HCM, it can be mid cavity HCM, or it can be classical, that is asymmetrical septal hypertrophy, or sometimes we can have obstructive as well as non-obstructive, or sometimes there is a concentric LVH like complete hypertrophy of whole muscles like complete LV, mid, base, apex, both septum as well as posterior wall. Okay. Sometimes it present with obstruction, sometimes it cannot present with obstruction. Okay. So, what will be the change happen in this pathology? Now, we just compare these two diagrams. Here LV cavity is large, here LV cavity space is now small. Now, this thickness is increased, this thickness is less. You can compare it from a bucket. Okay. A bucket which when you fill water, okay. So, center part is cavity and the wall of the bucket is muscle. Okay. So, now what will happen, because this space is lesser, okay. So, small amount of blood will enter inside LV cavity at the end of diastole, which we called as end diastolic volume. Suppose 100 ml blood will be entering at the end of diastole. So, here because cavity is small and here wall thickness is more, so pressure will be more. So, suppose here to start with, I told initially I have discussed, because of pressure difference blood will enter from LA to LV. So, in hypertrophic cardiomyopathy there will be less pressure difference because lower part is hypertrophied and LV cavity is small. So, more pressure is there in lower side and less pressure in higher side. So, when mitral valve will open, small amount of blood will enter into LV cavity and more amount of blood will remain into LA. So, you can imagine with subsequent cardiac cycle blood keeps on accumulating in LA. That is why over the period what will happen, LA will get stretched, it will start dilating, because amount of blood will enter, less amount of blood will enter in LV. Why? Because of this, because of small LV cavity as well as because of myocardial disarray, what will happen, the relaxation of LV during diastole will be lesser. The LV becomes stiff, because of stiffness it will not stretch, okay. So, one thing, LA keeps on dilating with subsequent cardiac cycle. So, blood will keep on accumulating with subsequent cardiac cycle. So, what will happen, these are pulmonary veins, these pulmonary veins will go back to lungs. Actually these pulmonary veins are connected with pulmonary capillaries and these pulmonary capillaries ultimately pulmonary artery. Okay. So, from pulmonary artery RA to RV, blood will come, RV to pulmonary artery to pulmonary capillaries and then to veins. So, if there will be traffic jam in this area, so congestion will be there in this area, and we know once congestion will be there in capillaries, the lungs will be filled with water, which we call as pulmonary edema or congestion. That is why these type of patients will have cough, okay, because alveoli is a closed space where gaseous exchange takes place, and when this membrane become wet because of this increased pulmonary congestion, okay, so the gaseous diffusion will decrease and patient will become dyspneic. There will be difficulty in breathing. Okay. Just a correlation how this pathology works, and then one most important thing which we have, why I am discussing all these things, because the discovery of drug are based on these things only, because once we understand the pathophysiology, we will hit at that point as a mechanism. Okay. So, now second thing, during systole I have discussed what will happen, these muscles will contract and blood will pump from LV to LVOT to aorta. Okay. So now what is happening here, because cavity, LV cavity is smaller and end diastolic volume is lesser. Okay. So at this time when these muscles contract, so when blood will come and enter to aorta, so at this point, that is LV outflow tract, this is left ventricle cavity and this is outflow tract and this is aorta. So when this small LV cavity with less amount of end diastolic volume, like less blood will go after contraction, so the velocity with which the blood will enter in LVOT will be very high. So as we have read in physics, when velocity will be very high, pressure will decrease. So at this point of time, what will happen? Pressure will decrease. Pressure will decrease in this area. Pressure will decrease in this area because velocity will of jet of blood will be very high. So because of decreased pressure, what will happen? What is the difference happening here? Here anterior mitral leaflet is closed. Here I think it's yeah. I adjusted it. So here we have discussed during normal physiology what will happen when LV will contract, both leaflet will close and whole amount of pressure will be applied here and blood will pump into aorta. But here what is happening, because of increased velocity, because of decreased amount of end diastolic volume, what will happen, because of Venturi effect, here at this area negative pressure will be created and this negative pressure will suck this anterior mitral leaflet towards outflow tract. So what will happen, blood will unable to go because now this anterior mitral leaflet will come in way in between aorta as well as LVOT and causing obstruction. So that is why after contraction blood will unable to enter into aorta because of less amount of diastolic volume and increased contraction of LV cavity because of hypertrophy. The velocity with which blood is entering in LV outflow tract will be very high. So velocity will be more, so pressure around that velocity jet will be very low. So because of negative pressure, there will be a suction pressure will be created here, and rather than closing and rather than this AML getting closed like this, it will just trapped here and causing obstruction at the level of door. So because of this, what will happen, blood will not enter to brain. It will not enter to aorta, it will not enter to brain. So that is why patient will have chest pain, patient will have giddiness, blurring of vision, syncope which we say, okay, sudden transient loss of consciousness. Okay. So now two most important thing is we have to focus on this. So lesser will be the end diastolic volume, smaller will be the LV cavity at the end of diastole. Okay. So more will be obstruction during contraction and more will be symptoms. Okay. So depending upon this, how we will treat this patient. So whichsoever drugs are discovered and made. So if what we have to do, we have to decrease LV contraction. So we will use those drugs which will decrease LV contraction. So the velocity will decrease, so there will be no negative pressure. Second thing, we have to increase end diastolic volume. So those drugs which will increase end diastolic volume, it will again increase the dimension of LV cavity. So the crux is, if diastolic volume will be low, if LV cavity will be smaller, symptoms will be more. And if LV contraction velocity will be more, again because of increased velocity, there will be obstruction of outflow tract and there will be more symptoms. So our end point is, we have to discover those medicines, okay, which decrease LV contraction and which increases end diastolic volume and increases LV cavity dimension. So now I will be covering in short regarding this investigation, because management in itself consists of investigation as well as treatment. First we have to investigate and then we have to treat. Okay. So during investigation there are sets of tests which we have to do. Okay. But clinically, see, I will not go into detail, but this is a 12 lead ECG, this is lead 1, lead AVL. So prominent Q waves are there in hypertrophic cardiomyopathy in lead 1, AVL, V5 and V6. and these are P wave. Normally these P wave are only prominent. There is no negative, there is no negative depth in this V1. So this negativity plus and then negativity in itself indicates that there is an increased LA pressure. So we have discussed because of with subsequent cardiac cycle, the blood will accumulate in left atrium. So over the period there will be dilatation of left atrium and there will be increased pressure in left atrium. So because of increased pressure in left atrium, there will be this increased negative part of P wave. Okay, this is an indirect indication of increased LA dilatation. Then normally this QRS complex are not so high. Okay, it's very high, 1, 2, 3, 4, it's almost 30 voltage. So normally it's half of that. So again, more will be the thickness of muscle, more will be the voltage. So it's an indirect indication of all these things. So this is how we can correlate clinically. Now this is ECG of apical HCM. Apical means lower part, this part. Means if HCM is in this area, so what will happen? Whole of the ECG, here we can see this T wave are positive, T wave are positive in V4, V5, V6. Even in, these are precordial leads. Okay. So here negative T waves in all leads. Okay, I will not go into detail because we also took so much of time to understand these things. So in fraction of minutes, it's difficult to get. So now this is echo. These are sets of tests we have to do. Okay, I'm just trying to correlate because once you will see like the gross anatomy, so you should be able to differentiate like which is whether the septum is hypertrophied or not. Okay, so this is echocardiography image. Okay, this is one view. Okay, see, you can consider this as a horizontal. Okay, now it's vertical. In there it was like horizontal, and here it is inverted. Okay, so we can correlate it here in a better way. Okay, so this is inverted structure. So we can see this is thickened structure. So this is septum part. Asymmetrical septal hypertrophy. This is very thickened. So basically in echo we need to differentiate with which pathology we are dealing with. So there can be so many closest differential diagnosis for discussing all these things. Here also you can see this is a thickened structure. So this is also septum. Okay, and this is anterior mitral leaflet. So as we have discussed during contraction, what will happen? These anterior mitral leaflet will move upward. So blood will not enter from LV to aorta. Okay, because the jet and velocity will be very high. Okay, so in echo, 2-3 parameters are very important. We have to see the thickness. This thickness is very important. If this thickness of septum is more than 30, then it's an indication for pacemaker. Okay, then LA enlargement. If LA is hugely enlarged, then again it's an indirect indication for going for surgery. Then we have to check mitral regurgitation. Normally I told you that blood will come from LA to LV and then from LV to aorta. But normally in HCM what will happen, because anterior mitral leaflet is closing here, so this area will remain vacant, means mitral leaflet are not getting close, AML is going ahead. So during contraction what will happen, there will be backflow of blood from LV to LA, which we called as mitral regurgitation. So all these are features which we have to see. See, again same structure, LV, LA, aorta. Okay, so blood will come initially from LA to LV, it has to go from LV to aorta. So blood is coming from, while contraction what is happening, blood is going to aorta but is going to LA also because of anterior motion of the mitral leaflet. Okay, so similarly we have to check multiple gradients. Okay, gradients mean when blood will go from LV to aorta. So because of obstruction, because of anterior mitral leaflet, the pressure will increase at this site. So this pressure is indirectly indicated like this. You can see it's a dagger shape if you see. So it's a like term there. I'm discussing all these things just as a sensitization, okay, or just for understanding. I'll skip all this part. In every diagram, so these are very important features. So if you see any patient with these features, and indirectly if you are doing some genetic testing, so these are indirect correlation for severity. Okay, these. So as we have discussed, the thickness of septum, which I have discussed in previous slides, more than 30 means the severity is more. Similarly, the dagger shape gradient which I have discussed, which indicates that there is some obstruction at the level of LV outflow tract because of jet of velocity, okay, velocity jet. So, if it is more than 30, then again it is very significant. Similarly, LA diameter I have discussed, with subsequent cardiac cycle blood will keep accumulating, there will be MR, so LA will dilate. So, all these are the severity index for aggressive treatment as well as for early interruption. So, next set of, so basically in every patient we do ECG, we do echocardiography for correlating everything, and sometime if we are not getting clear cut picture, but clinically we are suspecting, then that condition we have to do cardiac MRI. I will discuss the importance of cardiac MRI in subsequent slides. So, genetic testing I will not go into detail, now I will directly jump into my topic. Okay, management, how will proceed, how will manage these types of patient. So, again management depends upon, first we have to assess the risk factors. Okay. So, because of this myocardial disarray, because of matrix deposition, what will happen? So, see, we have conduction system in muscle, okay. In whole cardiovascular system there is a conduction system, SA node, AV node, His bundle, Purkinje fiber, everything, okay. So, ideally current flow in that in the form of action potential, which we read, action potential generation and then action potential propagation, okay. So, because of this disarray, what will happen? There will be abnormal depolarization and early repolarization takes place, okay, at the level of myocardium because of influx of sodium and outflux of potassium, because of change in membrane potential. So, because of all this change in membrane potential, so there will be dispersion of repolarization across those myocardial disarray cells. So, these myocardial fibrosed tissue in hypertrophic cardiomyopathy are prone to form or to throw arrhythmias, which can be either atrial arrhythmia, means coming from upper part of heart, right atrium or left atrium, or can be from ventricles also, which we called as ventricular tachycardia, ventricular tachyarrhythmia. What exactly happens in arrhythmia? Heart will beat very fast and there will be like the diastolic filling time will be very low. When diastolic filling time will decrease, so what will happen? Blood will not come. During systole, very less amount of blood will be there inside heart. So, outflow will be, cardiac output will gradually decrease. Okay. So, patient can get cardiac arrest. Okay. So, suddenly these type of patient, the first presentation of these type of patient are in athletes. Most of the time these patients are, if you hear like a basketball player have sudden cardiac arrest in the ground. Okay. So, the commonest cause is 60% time it is hypertrophic cardiomyopathy. So, the basic reason behind this is because of disarray and matrix dispersion. Okay, there will be dispersion of repolarization leading to throwing of arrhythmias, and these ventricular arrhythmia, what will do? When ventricular arrhythmia will be there, so heart will stop beating and patient will have sudden cardiac arrest and followed by sudden cardiac death. Okay. So, how we will assess the risk that which patient, which hypertrophic cardiomyopathy, which HCM patient will require pacemaker, or in other form, actually pacemaker therapy we do defibrillator therapy. You must have seen in movies that shock is given, shock is delivered, like they shoot a lot like this, giving shock. Yes. So, similarly what these in this HCM patient, whenever there will be ventricular tachycardia, so they can land up into cardiac arrest. So, we put ICD, that is cardioverter defibrillator. So, there is a battery here which we put in pectoral muscle, just above pectoral muscle, below subcutaneous fascia, okay. And then we attach lead, either RA lead or RV lead. So, two leads are there, one in RA and one in RV, and that RV lead have coil. So, these coil will deliver shock just like we deliver externally. So, these are made to sense ventricular arrhythmia, and once these ventricular arrhythmia are sensed, then these battery will sense, there are software which sense the abnormal rhythm. So, once normal rhythm is going on, it will not get activated. Once abnormal rhythm will come, this battery which we keep here, it activate and sense that VT is going on and heart is about to stop. So, immediately it delivers shock, 100 joule, 20 joule, 40 joule, whichsoever is set. We have all protocol in pacemaker. Okay. So, it deliver and then the rhythm gets reverted. So which patient are prone to develop ventricular tachycardia, which patient are not prone to develop? So we have to do risk assessment. So for that risk assessment, we have to take certain history. So there can be primary prevention or there can be secondary prevention. So suppose an HCM patient had cardiac arrest, had a history of cardiac arrest or had sustained ventricular arrhythmia, then that comes under secondary prevention, means he or she is the candidate to undergo ICD. ICD means we will put this cardioverter defibrillator. Okay. Then primary prevention means suppose patient has never ever have any episode of either cardiac arrest or ventricular arrhythmia, but if there is a strong family history like first relative history or first generation, if there is a history of sudden cardiac death in family because of HCM, then it is an indication to put ICD. Okay. So these are the risk factors. There are 4 or 5 risk factors. I have discussed just now the thickness of septum. If thickness of septum is more than 30 mm, then it is again a risk factor for ICD. If there is a recurrent syncope, बार-बार अगर चक्कर आ रहा है, बेहोश हो रहे हैं, transient loss of consciousness in HCM patient, it's again a criteria for putting ICD, strong family history. And if we do cardiac MRI, and cardiac MRI, if cardiac MRI is showing some fibrotic element, okay, which we can pick by late gadolinium enhancement, that is a feature. So basically you do cardiac MRI, and if cardiac MRI is showing that more than 50% of LV mass is fibrotic, fibrosis is because of this myocardial disarray only, secondary to all genetic dispersion. Okay. So all these are risk factor for ICD. So this is a protocol which we follow normally. So if patient is having prior event of ventricular arrhythmia or sudden cardiac arrest, then directly go for ICD. If out of these features which we have discussed, that is unexplained syncope, EF means ejection fraction. Ejection fraction means suppose 100 ml of blood will enter in heart at the end of diastole, and during systole what will happen? If suppose 60 ml of blood is going out, okay, so then ejection fraction is 60, means out of 100, 60% blood is going out. Okay. So that is normal, 60% is normal. So if EF is less than 50%, that is also an indication for ICD in HCM patient. So either one of this feature, if it is present, then we will advise ICD. If this is not present, if there is event of non-sustained ventricular arrhythmia, then again it is an indicator for ICD. If not present, we will do cardiac MRI. If cardiac MRI shows significant fibrotic burden, then again we go ahead with ICD. Okay. These are all like American College of Cardiology guideline. Whatsoever I have discussed, they have written same thing. Okay. Now coming to drug management. So one aspect is, whenever any HCM patient will come, you advise to get assessed for risk factor for ventricular arrhythmias. So those sets of points which we have discussed, if they are fulfilling that set of points, okay, then we advise this patient to undergo ICD. Then second, now coming to drug, okay, so we have already discussed the basic pathophysiology in HCM. So what are the requirements? We have to decrease LV contraction and we have to increase end diastolic volume. Okay. So these sets of drugs are there. So beta blockers, then calcium channel blockers. Okay. So I will directly discuss mechanism, what beta blocker does. So this cardiac muscles, okay, which are present in LV and RV. So cardiac muscles have beta receptors. These beta receptors, when they get activated, so cardiac myocytes contract via calcium calmodulin mechanism, okay, via sarcoplasmic reticulum. All this calcium system, they will, like the molecular level thing I am not going to discuss, but ultimately what will happen, because of rhinolidine receptors, because of T type and L type of calcium channel, okay, because of action potential, when these systems get activated, okay, so sarcoplasmic reticulum will throw calcium in sarcoplasmic, and there will be tropomyosin, troponin, actin troponin, everything that physiological changes, and there will be actin myosin contraction. So that is how, when actin myosin will contract, LV will contract. So ultimately this beta blocker are responsible for start of this process. So when we give beta blockers, okay, different type of beta blockers, so it will decrease LV contraction. Once LV contraction will decrease, what will happen? So outflow velocity will decrease and there will be less pressure, negative pressure creation across LVOT. So by giving beta blocker and calcium channel blocker, ultimately we are reducing outflow tract gradient, outflow tract pressure, and we are reducing MR also, because when pressure will be less, so there will be no anterior mitral leaflet movement. So anterior mitral leaflet will close, MR will be less, so LA pressure will also decrease and outflow tract gradient will decrease, so cardiac output will increase and backflow pressure will decrease. So that is how these drugs work. Calcium channel blocker, then calcium channel blocker, then there are other sets of drug, disopiramide. So disopiramide comes under antiarrhythmic drug. So it also acts, but its action, mechanism of action is by inhibiting. So in action potential curve, okay, so I will not discuss action potential, but the action potential curve consists of 0, 1, 2, 3, 4 phase, okay, because of sodium influx, because of potassium efflux and calcium influx, like that. Okay, so these medicines basically, see, for contraction what is required? It's very simple logic. For starting a fan or light, when we switch on button, what happen? In that wire, current will flow and then it will blow up the light. Okay. Similarly, what happens in our cardiac muscle? Cardiac muscles are supplied by conduction system. So these conduction system will throw current. How? In the form of action potential. So when this action potential travels via conduction system into myocardial muscle, okay, it will cause all calcium release and contraction. So this action potential generation will be inhibited by disopiramide by inhibiting sodium channel absorption, okay, by inhibiting sodium channel receptors. Now for each group of patient we will not, yeah.
So can you re-explain the metoprolol, which is a beta blocker?
Yeah.
Re-explain the mechanism of action of that, how it affects
Yeah.
the ejection volume.
Okay. So basically, ma'am, what we are discussing, because of LV contraction, what will happen in hypertrophic cardiomyopathy? LV cavities is small, end diastolic volume of blood will be less. So in this condition, when heart will contract, so the velocity with which blood will enter into aorta will be very high. So because of that, at LV outflow tract, because of Venturi effect, what will happen? The negative pressure will be created around jet of blood. So because of that, there will be this anterior mitral leaflet will come close and obstruct the outflow tract opening. So because of that, what will happen? The normal path which is open, okay, after mid systole, it will get closed. So that is why that's why blood is not able to enter into aorta. So we have to decrease LV contractility. Okay. So myocardial muscles have receptors. They have beta 1 and beta 2 receptors. These beta 1, beta 2 receptors, when they get activated, so they will activate calcium calmodulin system of myocardial muscle, which once get activated via rhinolidin receptors are present. Okay. So these rhinolidin receptors will activate calcium calmodulin system, and there will be secretion of calcium into the sarcoplasmic reticulum. These calcium will enter and it will attach with troponin T and troponin I, and tropomyosin will uncover, and then actin and myosin will come close and they will contract. And when actin and myosin filament contract, there will be contraction, because group of myofibril will contract, so group of muscle will contract. So group of muscle will contract, so LV is made up of muscle. I told you initially only that the LV is like a bucket. Okay. So when this wall will contract, so because it is a cavity, so blood will forced out. Okay. So if beta blockers means we give drugs which inhibit this beta receptor action. So we inhibit beta receptor action. So we inhibit actin myosin contraction indirectly. So lesser will be the contraction. So blood will enter into the LVOT with less velocity. So more negative pressure will not be there. So the suction of anterior mitral leaflet will not be there. So there will be no obstruction. So blood will enter easily. because gradient which I showed indirectly in echo as a dagger form, it will be very less than 30. And because now anterior mitral leaflet is normally closing, so mitral regurgitation, that is some amount of blood which was entering from LV to LA will be decreased. So LA over the period with subsequent cardiac cycle, what will happen? LA pressure will also decrease. So pulmonary congestion in pulmonary capillary, everything will decrease. So that's how, like, for understanding mechanism of these drugs, so that's why I have discussed that normal and abnormal physiology. Okay. So all drugs, that's why there are, see, if you see HCM patient, no? So HCM patient, when they stand, okay, when they stand, so they feel giddiness or they feel syncope, they feel transient loss of consciousness. So there is everything is because of physiology. So normally when we stand, 500 to 600 ml of blood will, the venous return. So normally venous return means amount of blood which is entering in our heart via veins, inferior vena cava. So when we lie down, 500 to 600 ml of blood will extra amount of blood will enter into heart. And when we stand, because of gravity in calf muscle, you must have seen, because of standing, the calf muscles veins get dilated and varicose vein, because calf muscle have venous capacitance. Okay, so they can stretch. That venous system have property to get stretched. So when we stand, 500 to 600 ml of blood will pull in our leg. So the venous return will decrease. When venous return will decrease, you can imagine 500 ml of blood is decreasing in HCM patient when they stand. So end diastolic volume will decrease. So again, what will happen when end diastolic volume will decrease? LV cavity will become small. Now again, when muscle will contract, the velocity will increase, and again there will be more suction of anterior mitral leaflet. There will be more obstruction. You just advise this patient, okay, to take good amount of water, increase fluid only. So that's why we avoid, like the instruction which we give to this patient is avoid dehydration, avoid taking diuretics. Diuretics means those drugs which pull out water, which pull out water via urinary system. Okay, basically it decreases venous capacitance. Similarly, if you ask this patient to do Valsalva maneuver, Valsalva maneuver means you have to close your nose and mouth, and you have to blow against closed glottis. Glottis means basically you have to close your mouth and nose, and you have to blow against closed mouth and glottis. Okay, so what will happen? Your intrathoracic pressure will, your chest will expand, so your intrathoracic pressure will increase. Okay, so our heart is like this, okay, and lungs are like this. So when we will blow like this, so intracavitary pressure will increase. So it will again, when cavitary pressure will increase, it will transmit to pericardium. Pericardium is layer around heart. So our cardiac chambers will compress from outside. So again, end diastolic volume and LV cavity will decrease. So if you ask HCM patient to do Valsalva maneuver, they have transient loss of consciousness, because immediately what will happen? Outflow tract will get obstruct and blood will not enter to brain, and immediately they will feel loss of consciousness or transient loss of consciousness. They will fall down immediately, and the moment they fall down, the venous return 600 ml blood will go and they will regain consciousness. So like that. So that's why our treatment is, we have to increase end diastolic volume, we have to increase LV cavity dimension, and we have to decrease LV contraction. So all those drugs basically decreases LV contraction. We have to avoid giving diuretics, but still diuretics are given in HCM patient. There are few indication. Suppose patient is in heart failure. Heart failure means lungs are wet. Okay, lungs are wet because again same thing. LA is dilated, LA pressures are high, pulmonary capillary wedge pressures are high. Okay, so lungs are wet. Alveoli is a space with membrane where gaseous exchange takes place, but when they are wet, there will be no diffusion. Okay, only carbon monoxide gas diffusion is very high. CO2 and oxygen diffusion is low. So it will get effect, and then less amount of oxygen will reach to brain. So patient will become dyspneic, air hunger. Okay. So because when we increase respiratory rate, so there will be forceful inspiration and expiration. So more amount of blood oxygen will enter. So that's how they are able to manage. So that's why they will sit like this. Okay. forward bending position, trapping position, because in this position what will happen, your sternocleidomastoid muscle, scalenus muscles, Eller nasi, all these help to inspire more. So this is a typical, by standing from like far away you can make out that this patient is having dyspnea and he is landing into failure. Okay. So that's why like everything is correlated with physiology only, and if you will understand physiology and pathology, you can correlate the symptoms. So now all group of drugs, there are other new sets of drugs which I will discuss. They inhibit cardiac, this myosin inhibitors, actin myosin contraction, so they just inhibit myosin ATPase enzyme. So they inhibit myosin ATPase enzyme, so there will be less contraction, because there will be less actin myosin overlapping by inhibiting myosin ATPase enzyme. Okay, so that's how again what we are doing. By doing that, we are again decreasing LV contraction. By giving beta blocker, we are decreasing LV contraction. Calcium channel blocker, they directly act on that calcium calmodulin receptor, and they inhibit the transition at the level of ranolidine receptors. Ranolidine receptors, like it's a very interesting thing, but it will take whole day to discuss. But just I am trying to understand the gist so that you can understand. I hope they have learned physiology, physiology. I know I did BSc 2 second year. After that I selected. So I remember we used to do muscle twitching, and prawn, I used to dissect prawn and frog, everything. So means basically the crux is muscle twitching in itself. If you are learning muscle twitching and everything, you should understand action potential, neuromycin receptor, everything, acetylcholine and everything. Yeah. Yeah, so diuretics means diuretic means urine output is increased. Okay, some amount of blood which is accumulated in pulmonary capillaries, okay, that is thrown out of body via micturition. So one more thing, so dehydration should be avoided in these patients, same thing. So that's why HCM patient, like during our PMT times or even PG entrance time, we used to get questions like, a young athlete, basketball player had sudden cardiac arrest, what is your diagnosis? So most commonly 60% time it's hypertrophic cardiomyopathy only. The reason is because when you play, you sweat a lot. Okay, so sweat means dehydration. Dehydration means venous return will decrease. Again, when venous return will decrease, what will happen? LV cavity will decrease. Okay, same physiology, everything. Similarly, one more thing happened at that time. What will happen? There, when we run, so there will be vasodilatation. Vasodilatation means our capillaries or our arteries have a property to contract and expand. Okay, so when you run, so your muscle should require oxygen for your mitochondria to get that all process, oxidation and respiration. So for that oxygen is required. Okay, so for entry of oxygen at that level, all peripheral arteries will get dilate. Okay, so when peripheral arteries will get dilate, what will happen? Similarly, this aorta, okay, means from LV to LV outflow tract, then aortic cusp, then aorta. So if aorta will get dilate, so what will happen? When aorta will dilate, so the pressure against which LV will contract will decrease. So again velocity will increase. Okay, see, if you want to throw some, like if LV will contract and LV has to push blood to aorta, okay, and if aortic pressure is very high, so the velocity will be low. But if aortic pressure is low, so the velocity will be high because resistance is less. So same thing happens in like athlete. They are running, so vasodilation will be there because cardiac output requirement has increased 2 to 3 times. So aorta will dilate. So afterload, we say it as afterload. Afterload means the pressure, amount of pressure applied by aorta for left ventricle or against left ventricle to pump blood. So during this condition, what will happen? LV will pump against less pressure, so it will pump more. So again velocity will increase. So velocity will increase and end diastolic volume is decreased because of dehydration. That's why these athletes, they have sudden cardiac arrest, because gradient will increase high. Subsequently, like normally the gradient across LV OT, okay, LV outflow tract basically. So the gradient across LV OT, gradient means pressure gradient. See again pressure gradient also I will explain. this is LV, then this is LVOT, outflow tract, and this is aorta. So because anterior leaflet will suck here at the time of mid systole during contraction. So now LV has to pump blood in aorta in any condition. So at the mid of systole, what will happen? He got or he or she, I don't know, he or she will got the obstruction there. Okay. So LV has to overcome this obstruction. So he will contract in like very rigorously. So pressure will be very high here. Okay. That's why with this pressure, suddenly there will be opening. Okay. But this pressure gradient is causing symptoms, because this sudden obstruction will lead to, for fraction of second, blood will not enter in brain. For fraction of second, what will happen? From aorta, base of aorta, coronary arteries are there, and these coronary artery in itself supply myocardial muscle, because of which heart attack happens. This coronary artery which got blocked, okay, for which we have to do angiography and angioplasty. So basically it arises from base of aorta. Okay. So when this get obstructed and blood will unable to enter in aorta, so coronary will also get less supply. That's why when supply will be less and demand will be more, so patient will have pain. So that's why patient will have syncope, chest pain, okay, and breathlessness. This is the drug about which I was talking. This is a new drug. Recent trials are going on, Pioneer HCM trial and Maverick trials. Mavacamten. So mavacamten is myosin 80-pase inhibitor. So ultimately what it's doing, it is also inhibiting LV contraction. Okay. So I'll take just 5-10 minutes more. Okay. I'm so sorry, the topic is like that. I'm trying to explain in it's very difficult to understand also, but once you understand, it's very easy also. It's like so I'm trying to like discuss it or like to dissect it so that the physiology and pathology should be clear. The gist is to understand, because every time we can go back and study and read, but if you understand the concept, then you'll never forget all these things, and everything you can correlate clinically. So once again, whenever you have these patients, okay, we have to assess risk factor for ventricular arrhythmias. Okay, certain sets of risk factor we have discussed. If patient will fulfill either one of those risk factors, he or she is a candidate to undergo ICD. ICD means, we have already discussed, just like external defibrillator, ICD is an internal defibrillator. There is a battery, there is a wire. I'll show you, I have one video outside. Okay, so we attach one wire into RA and one wire into RV. Next speaker is there, can I? Okay. Now, okay, sir, thank you very much. So then subsequently drugs are discovered on the basis of this concept only, which we have already discussed in detail. Okay, most of the drugs are acting by decreasing LV contraction only. Okay, now few drugs which we have to avoid giving in this situation is, one we have discussed, diuretic, because again diuretic will cause dehydration, it will cause decrease end diastolic volume. One more drug is AC inhibitor or ARB, angiotensin converting enzyme inhibitor and ARBs. Okay, angiotensin receptor blocker. Basically these are vasodilators. So just now I have discussed, if you will give vasodilator, so what will happen again, the force velocity will increase, so Venturi effect will be more. Okay, so AC inhibitor, ARBs, diuretic has to be like, these are contraindicated. But sometime what happens, so whenever these patient will come, how will escalate medicines? So first of all, not all HCM patient will require drug. Okay, if HCM patient is having obstruction, so there is a terminology you will see, obstructive and non-obstructive HCM. Obstructive HCM means anterior mitral leaflet movement is there because of Venturi effect and pressure gradient is there. Because of that, patient is symptomatic. So if pressure gradient will be more than 30, that dagger shape in echo which I have shown. Okay, so if pressure gradient will be more and if patient will be symptomatic, to be specific, if pressure gradient will be more than 30 and if patient is having dyspnea means breathing difficulty, chest pain or syncope. Syncope means chakkar, giddiness. So then he or she is a candidate to start with medicines. So normally we will start with beta blockers, okay, either carvedilol, metoprolol, nebivolol. Okay, these beta blocker we try to increase drugs to the maximal tolerated dose. In spite of that, if these patients are having symptoms, we add second group of drugs, that is calcium channel blocker, diltiazem, verapamil, like these group of drugs. Again we will give this combination therapy, and if these patients continue to remain symptomatic and gradients continue to remain high in spite of giving these, then we will add these group of drugs like myosin inhibitors or disopiramide, that drug which inhibit action potential, which block action potential, okay, sodium influx. In spite of that, still if patient is symptomatic, then he or she is a candidate for surgery. Then again in surgery there are two options. See, it is a very simple thing. Again surgical concept is also same. Why everything is happening in LV outflow tract? Because of asymmetrical hypertrophy. Okay. So why surgery? What we are going to do? That extra amount of bulging we have to remove it. So either you have to cut that extra part. When that extra part will be cut, what will happen? Every Venturi effect and everything will go away. Okay. So patient will revert back. Gradient will decrease, no Venturi effect, no MR, decrease LA pressure, everything. No, ma'am. There are two. I am coming to that. So one is open heart surgery, which we called as Moss procedure. Okay. So which we call as myomectomy. Okay. Directly we have to cut that part. Okay. But it's an open heart surgery. Okay. Then other option is, if patient is not fit for surgery, then as a cardiologist we can do alcohol septal ablation. Alcohol means absolute alcohol. Okay. That 100% alcohol, 1 to 3 ml of alcohol we have to put in septum, septal branch. Just a minute, I'll just for understanding, I'll see this is the same diagram. Sorry, I am repeating again and again. I couldn't get time to like prepare more. I am so sorry for that. So see, this is partner. So here incision will be taken. Okay. So via aortic approach. So normally chest is open, okay, chest is open and then all pump is attached, cardiorespiratory pump is attached to heart, and then via aorta, okay, you take an incision in aorta, okay, open aorta, and via aorta you will be able to see interventricular septum. So you can see your eyes here, suppose your eye is here, you have given incision here, okay, and then you will be able to see this septum. So you will be able to see this bulge, okay. So now you go inside and you will cut this part. Once this part will cut, again it will revert back to this position. So all the Venturi effect will be gone. So patient will be symptomatically better. Gradient, Venturi effect, anterior mitral leaflet, MR, everything will. Over the period, 10 to 20% chances are there that this LV cavity will also grow. Like means the cavity which has become shorter, it will expand slowly and the pressure will come. Most of the time this LA takes time, okay, to regress, because there is something called myopathy. Again atrial myopathy also takes place, but that is like the fate of patient. So like this, and then other thing is, if so when you will prefer surgical ablation, that is myomectomy, we have already discussed. After giving GDMT, GDMT means guideline directed medical therapy. Whatsoever we have discussed, beta blocker, calcium channel blocker, disopiramide, then myosin inhibitor, all these group of drugs, in spite of giving that, patient is symptomatic and patient is having increased gradient, then we prefer this patient either for alcoholic septal ablation or surgical. So if patient has associated abnormalities, for example, suppose see, now again just focusing on more structure, these are anterior mitral leaflet, posterior mitral leaflet, these are chorda tendini, these are papillary muscles, okay, and this is aortic wall. So if patient has associated aortic wall stenosis means aortic wall is not opening. Over the period what will happen, sometime in because of calcium deposition this wall gets stenosed, okay, just like you can imagine door of home. Over the period when it get rusted, okay, so the opening will be restricted and there will be sound. Same thing happened in this door also, okay. So when it is unable to open, again the gradient will increase and everything. So patient will have severe aortic stenosis. So if this both thing, severe aortic stenosis with this, so in spite of going with alcohol ablation, we will prefer it for the surgical procedure, because at a time both pathology will be taken care. Similarly, if patient have internal mitral valve disease, if patient have anomalous papillary muscle, if patient have abnormal elongated mitral valve, all this disease, or suppose if patient has, what I was talking, see from here coronary artery rises, from here and from here. So suppose patient is having coronary artery disease, patient had underwent heart attack also, if after doing angiography patient is having triple vessel disease, there are 3 coronary arteries, suppose all coronary arteries are blocked and patient is having HCM also. So these patient directly rather than doing septal ablation will shift this patient for or prefer this patient for surgical procedure, because in same sitting bypass can also be done, means grafting can also be done, and simultaneously this pathology can also be taken care. Okay. So similarly there are few sets of condition, if patient is having severe pulmonary artery hypertension which is progressing, means just like from LV aorta arises, from RV pulmonary artery arises. So if there is a severe pulmonary artery hypertension because of increased LA pressure, we were discussing the LA pressure will increase and pulmonary congestion will be there over the period for 20 years, 25 years, if patient will not take anything. Okay, so over the period everything will get, just like a thing like if you will get traumatized here, okay, and if you leave it like that, over the period what will happen, scar will form, okay, and it will be deformed, taut. Same thing happened with everything in body. Okay, over the period if you will neglect, so everything will get distorted. Okay. So that's why over the period because of MR patient can have persistent pulmonary artery hypertension. Okay. Then again it's an indication for surgical myomectomy. Okay. Then if the gradients are in spite of 30, if the gradients across LVOT will be more than 100. Okay. And if patient have arrhythmia, LA is hugely dilated, means in short if there are multiple associated structural problem along with that, then better to go with surgical. If patient is not fit for surgery, severely uncontrolled diabetes, morbidly obese, metabolic syndrome, dyslipidemia, everything, okay, it's very difficult that patient will come out from ventilator. So in that condition we prefer for alcoholic septal ablation. In alcoholic septal ablation the basic concept is, see this is coronary artery, we were discussing that from here aortic root, coronary artery arises. So this is one coronary artery, okay, this is called as LAD, left anterior descending artery. Left anterior descending artery will give one branch called septal branch. Okay. So this septal will supply septum. So the concept is, see, just like each and every tissue requires oxygen, similarly cardiac muscle will require oxygen for doing all metabolism. So from where they are getting? From these arteries. So this artery will give septal branch. The name is septal because it is supplying septum part, and this septum is now hypertrophied. So what we'll do, we'll go inside, okay, and we'll give 1 to 3 ml of alcohol here and we'll occlude this branch. So by giving alcohol what we are doing, we are occluding that branch. So once branch get occluded, what will happen? Blood will not enter, and over the period what will happen? Tissue will get necrosed. Okay. So over the period what will happen? Automatically tissue will get necrosed. So this bulging part will get necrosed, and over the period it will get fibrosed and it will regress. Once it will regress, again outflow tract will be not there. Okay. Just like what we are doing via surgical, we are doing via internally, but it takes over the period some time. There are some drawbacks with this method because success rate is 80% with this, success rate is almost 95% with surgical thing. If there is spilling of alcohol in LAD, okay, mind you, patient will have anterior wall MI, like patient will have attack, patient will have heart attack. There are very less chances, but sometime all complications are sequel of our procedures that cannot be like handled by anyone, like this thing. Handled by anyone means like what is going to happen is not under our control, but if it has happened we try to control, and many times we can like manage to control. Okay. Other thing is, see, if you will see clearly, this is RA, this is RV, this is LA, this is LV, here is SVC, so is SA node. From SA node here, AV node, his bundle. So if you will see, this is area of his bundle. Okay. Here right bundle, left bundle, anterior fascicle, posterior fascicle. Sometime what will happen? there will be, because of tissue necrosis, sometimes this conduction system get damaged. So patient will develop block, heart block, complete heart block. So in 15 to 20% cases, patient may develop these two, complete heart block also. So now for complete heart block, again you have to see, this is pacemaker, this is a battery, one lead is here, there are two lead, okay, one lead will put here in RA and one lead will put in RV, okay. So this is like at pacemaker means it's coordinating all activity, because we have already discussed conduction system means action potential will travel, and this action potential when reach to myocardial muscle, it will activate and there will be contraction. But if there is a damage in area, okay, so now if action potential is unable to reach by a natural pathway, via pacemaker we are going to stimulate cells. So that is how it's we have to deal with this situation. So these are the comparison which I have discussed with myectomy and ablation. Okay, 20% chances of this AV block. High grade AV block means that same complete heart block, and with this procedure myectomy 2%. Success rate is 80%, here 90%. But one better benefit is that with ablation, alcoholic ablation, gradient will fall at least 20 to 25, like the outflow gradient, that dagger shape because of Venturi effect. So better control of gradient is with alcoholic ablation. Okay. This is a protocol. So same thing which I have discussed, like you have to obstructive patient with symptoms, go ahead with medical therapy, start with beta blocker, calcium channel blocker, disopyramide. Okay. If not responding, persistently symptomatic, these are candidate for surgical. Surgical we have discussed. If patient have associated structural abnormality, go ahead with surgical. If not, or if there is a contraindication for surgery, not a good candidate for surgery, go with alcoholic septal ablation. If patient is having prior history of cardiac arrest or ventricular arrhythmia, go ahead with ICD. If patient is having a positive history, family history of death in family history of death in relatives, first degree relative, okay, because of HCM, or if there is septal thickness is more than 30, if there is recurrent syncope, or if there is severe LV dysfunction, EF less than 50%, all these criteria, then go ahead with primary prevention. Okay, thank you very much for your patient listening. I hope I tried to discuss something.
Thank you so much, sir.
Yeah.
This was definitely an interesting talk. And before concluding, I actually request our HOD, Professor Narahari Gramapurit sir, to come up on the stage, Professor Sakthivel Sadayappan sir to come up on the stage, and course coordinator Varsha Vankade ma'am to come up on the stage and have a photograph session with our cardiologist, Dr. Abhinav Mohabey. Please, sir.
I'm sorry.
Richa ma'am, please come up on the stage. Thank you, Dr. Mohabey. This was really an interesting talk. We really got to know so many intricate details about our heart structure. And with this, we really want to go for a coffee break, and then we will again meet soon after 10 to 15 minutes, and we will have our session with Professor Sakthivel and Sadayappan sir. So, thank you, everyone, and we will meet shortly after a break.